CAR T cells are revolutioning the treatment landscape for several hematological disorders, including multiple myeloma, non-Hodgkin lymphoma, and B-cell acute lymphoblastic leukemia. And this is the main field of application so far of the commercially available product in childhood. But B-cell precursor ALL is not the only hematological malignancy. There are other malignancies that could benefit from the approach of CAR T cell. In particular, we invested on T-cell acute lymphoblastic leukemia, choosing as target an antigen called CD7. Unfortunately, this molecule is expressed on both leukemia cells and on normal T lymphocytes, and this could be associated with the risk of a reciprocal killing of the CAR T cell that express this antigen, this phenomenon being known as fratricide. To overcome this obstacle, we took advantage of a CAR construct in which there is a DNA sequence blocking the expression of the target molecule inside the cell, in particular in the Golgi apparatus and the endoplasmic reticulum. This means that the CAR T cells are CD7 negative. They escape the phenomenon of fratricide and they can kill the leukemia cells. We have treated already 20 patients in my center, thanks to a collaboration with the University of Singapore, in particular with Dario Campana, and with a small bio company that has this CAR construct. And we are now running a clinical trial with impressive and very promising results that in the future will be open also to other European academic centers. This option may be very useful, is another opportunity to reduce a state of remission in those patients who demonstrate to be resistant to the conventional approach. It's a sort of very effective rescue strategy for patients with relapse or refractory T-ALL, and it's a concrete therapeutic option already available in our continent thanks to the study that we are running. The use of the autologous CAR T cell is both before and after transplant, in the sense that it's a strategy for obtaining a complete remission in patients relapsing, and then these patients can be transplanted to consolidate the result. But it can be also an option for those children relapsing after allogeneic stem cell transplantation, because we can generate the CAR T cell if the patient has relapse not earlier than 100 days after the allograft, and also patient treated after a bone marrow transplant relapse had a very favorable outcome. In the clinical trial that we have opened, patients that can be enrolled must have an age comprised between 6 months and 25 years. And this is exactly the age range that was adopted also for the LIANA trial, which led to the approval of TISA-cel in both Europe and US. All the commercially available products are based on the use of autologous CAR T cell, namely CAR T cells generated using the patient's lymphocytes. But unfortunately, sometimes the patients can be extremely lymphopenic, and we don't have the opportunity to generate the autologous CAR T cell. An allogeneic product can be an alternative, which has several advantages because it is based on the use of T lymphocytes never exposed to chemotherapy or to steroid therapy. In addition, there is no risk of leukemia cell contamination in the collection of the cells that are then manufactured in the GMP facility. And the last point is that an allogeneic donor doesn't have also an increased amount of myeloid-derived suppressor cell or regulatory cells, potentially jeopardizing both the manufacturing and the function of the CAR T lymphocytes. At the very beginning, there were concerns about the risk of inducing graft-versus-host disease, or also about the possibility that the cells be rejected. But thanks to several efforts in different contexts, including acute lymphoblastic leukemia and even solid tumors, we have been able to demonstrate, and the results have been published in several papers, that these CAR T cells obtained from a donor are able to expand and persist into the patients, and the risk of inducing severe GVHD is negligible. It's important to invest on science, on research, and to focus efforts on the generation of more effective, more specific and selective therapies. And certainly CAR T cells have the potential to change the scenarios in other childhood disorders, including T-cell acute lymphoblastic leukemia, acute myeloid leukemia, and selected types of solid tumors. So I invite everyone to support research efforts to promote the advancement of science in the field of adoptive immunotherapy with CAR T cell.